Hey everyone, this is Brenda Pacheco. This is for Bio 225, and this is for week number 4. I'm doing exercise number 1, the methyl red and bogus prosquar testing. I'm not sure if I said it right. But anyways, so I have here my, I have labeled my, my MRVP broth with S. epidermidis and with the E. coli. So the first thing I'm going to do is open the E. coli broth, and I'm going to pass it over the flame to sterilize the tip. And I'm doing that as well for the S. epidermidis. So I'm going to grab my E. coli MRVP broth, and then I'm going to sterilize the tip as well. Okay, so once I did that, I am going to carefully grab my pipettes from my alcohol, and I am going to grab my E. coli broth here and grab a little bit of it. And it says to drop, I think it's 4 drops into the active culture of the MRVP tube. So 1, 2, 3, and 4. And then I'm going to place my pipette back in the alcohol, and eventually I'll put it in the bleach. So before closing this, I'm going to sterilize the tip of this again, of the active MRVP broth tube. And then now I can put the lid. So I will be doing the same thing for the S. epidermidis, and then I am going to incubate these tubes for 48 hours, and then I'll check back with you guys. Thank you. Hey everyone, this is Brenda Pacheco, and I am doing the experiment for week number 4. This is for Bio 225, and I'm doing here exercise number 2, the catalyze testing, for biochemical testing for microbial identification. So I went ahead and I labeled my slides already with E. coli and S. epidermidis, and then I have here my hydrogen peroxide and my alcohol soaking with the inoculated loops. So I'm going to first start with the E. coli, and I will be grabbing a sample of my plated culture here, and I will be adding it onto the E. coli here. So I'm trying to get a good chunk to hopefully see result. Jackie, don't move. And then I will be putting it on my slide here. Okay. And I will also be doing the same thing for the S. epidermidis. However, I need to add my 1 drop of the hydrogen peroxide on the E. coli slide. And there it goes. So I'm doing the same thing for the S. epidermidis. And I'm also going to add 1 drop to my slide. Perfect. So I am going to observe these samples for the formation of oxygen bubbles, and then I will be taking a picture of my slides. Thank you. Hello, this is Brenda Pacheco for Bio 225, and this is for week number 4. This is part 2 of the methyl red and bogus prosquour testing. So for the sake of time, I already grabbed my E. coli and S. epidermidis tubes, and I spread them in each of these 4 tubes here. So I got 2 S. epidermidis and 2 E. coli. So, okay. So I'm going to be opening up the methyl red and adding 7 drops, 1 in the E. epidermidis and 1 in the E. coli. So here we go. 1, 2, 3, 4, 5, 6, 7, and then 7 drops into the E. coli. 1, 2, 3, 4, 5, 6, 7. Okay. Now I'm going to be grabbing the Barrett's A reagent, and I will be adding 12 drops in each of these 4 tubes. And then after that, I will be shaking it up. Okay, so first I will be adding it to my S. epidermidis. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 more drops into the other S. epidermidis. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 into the first E. coli. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And lastly, 12 more onto the second E. coli. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So now what I'm going to do is I'm going to shake these. I am exposing them to oxygen. Okay, and then lastly, I will be adding the Barrett's B reagent, and I will be adding 4 drops into each test tube. So first into the S. epidermidis. 1, 2, 3, 4, and onto the second S. epidermidis. 1, 2, 3, 4, and then 4 drops into the E. coli. 1, 2, 3, 4, and then the last E. coli. 1, 2, 3, and 4. And then it says to shake tubes for 30 seconds. So again, I'm exposing them to oxygen. And while this went from a purple to a like mustard yellow, this is for the S. epidermidis. Now let's check the second S. epidermidis. This one didn't really change in color. It's just clear, like a clear white, like a cloudy white, I should say. Okay, let's see what E. coli does. It turned into like a dark yellow. And the last E. coli. So now that I have done this, it says to incubate my tubes for 30 minutes. So I will be leaving them like this, exposing them to oxygen. And then what I'm looking for is, I'm observing for color change. And if there is a color change, that means that a positive test is the formation of a red surface layer. So then I will be taking a picture of my tubes, how they turn out, and then I will check back with you guys. Thank you. Hey guys, so this is Brenda again, and this is part 3 of the Voges Prosquour test. I have been incubating these for a little bit over 30 minutes, and I noticed really not much color change. I'm not sure what went wrong or what I did wrong. I kind of actually noticed a little bit of change on the S. epidermidis and the one back here, both of the S. epidermidis. So I noticed that when I first was doing the test, these tubes were much lighter in color. It almost looked like this, but a little bit more clear. So I guess I could say that this one kind of changed color. But the E. coli ones, I didn't really notice much change. It's actually stayed the same in color. So I was hoping to see the red surface layer for a positive test, but I guess not. So this has been the end result of my test. I mean, you can kind of see a little bit of, I don't know, it's like a dark orange, but not really. I wouldn't say it's red. So yeah, that has been my experiment for week number 4. Thank you for watching.